All right, hello again, everybody. Oh yeah, so Robert, actually, a little bit of a history lesson for you. So back in the Middle Ages, like the um, superior form of warfare was mounted cavalry, right? Knights in armor and stuff like that. And typically, knights would be the kind of like the semi-useless sons of nobility, right? So they weren't, like the second or third or fourth sons. So they either went into the clergy or they became knights and went around chopping people up. And uh, what changed that was the development of the longbow, right? Which was, you know, probably I think they're about 7 feet long. These bows, 6 to 7 feet long, made out of willow, and they had sufficient range and penetrating power that they could actually kill knights. And also that was kind of like a massive upset in the social order, where because archers were peasants, or people who are working in the fields or serfs or whatever. And so this was a great British kind of invention. And uh, basically, longbowmen, and you had to be really bloody strong to pull one of these because they are big. Um, basically, longbowmen tipped the balance of superiority against the French in one of the many, many, many wars that the British had with the French. It was like pretty much constant for several hundred years. What's up, Jordan?
Did crossbows come into play at some point? Like, I don't know when crossbows were made, but I
That
always
was
figured they also did something.
Yeah, that was quite a lot later. Actually, crossbow killed Richard Lionheart. He got one in the eye, I think, from when he was laying siege to some French castle for some reason or other. So that was later on. That was kind of even more of a democratization, if that's the word, of firearms. Anyway, so the French hated British longbowmen so much because they had such range and power that whenever they captured a British longbowman, or English, I guess, at that point, they'd chop their fingers off. The string pulling fingers. And so, as legend goes, obviously I wasn't there, so I can't say. At the beginning of each battle, when they kind of stand off against each other, the English longbowman would go hee hee hee, as in I've still got my fingers. So in UK, that's an insult, kind of equivalent to that. But so I don't take offense. I really don't mind. It's totally cool.
Okay, good
But
thing we're in the U.S. So
yeah. But in the
it's
UK,
more of just peace out. I'm out of here
right,
type of thing.
exactly.
Okay,
In the
noted.
UK, you would be saying something rude to someone if you did that. But that's what it comes from. Uh, probably 700 or 800 years ago, in terms of like where that sign originated. Somewhat of an apocryphal story. I'm not exactly sure how historically accurate it is, but it's a fun one nonetheless. And then, as Jordan pointed out, obviously everybody got crossbows. It was like, yeah, whatever. Anybody could kind of shoot anybody. Didn't have to be all that strong. So what we're going to do now is we're going to take our good look at meiosis. So again, kind of always try and make sure that I give you a feel for the kind of the bigger picture, because I find that helps a lot with understanding. It's not just a collection of stuff. And so meiosis is all about randomness, because the purpose of meiosis is to create gametes for sexual reproduction. I'll write that down because that's important. And more importantly, haploid gametes. And another extra word there would help, for sexual reproduction. So sexual reproduction is costly, right? In many, not just talking about human behavior, but in kind of any, even plant sexual reproduction, right? Because for one, you only contribute half of your genetic information to the next generation, right? So in terms of kind of passing on your genetic information, asexual reproduction is the winner, right? Because all of your genetic information goes into the next generation and so on. Jordan.
Nice, sir. I know you talked about like inbreeding and whatever and whatnot, being negative because you get like you're more likely to get the negative traits. How does it work with asexual? Is it just is the likelihood the same or?
Yeah, so asexual reproduction is really good if you want to reproduce very fast, right? Because it's very quick compared to sexual reproduction. And also if you're in an environment that doesn't change. So if you're in a fairly static environment where the next generation is going to encounter pretty much the same conditions in terms of food, predation, so on, as current generation, then asexual reproduction is the way to go, right? It's kind of advantageous.
Okay, so the mutations that are, I mean, I guess depending on the flavor, they could be bad or good. So in this case,
yeah, it is a very interesting point about asexual reproduction because there's no purging of deleterious mutations, right? Which would occur either through DNA replication or, external insults. Give me just a second. I have to shout to one of my children. Hey, Evie, Sophie, can you let Luna back in, please? Thanks. Sorry, my dog's out. Well, one of my dogs outside. Want to get let back in. So, yeah, they do have. So, kind of getting off a little bit into the weeds, but some asexually reproducing organisms do have the option of going through sexual reproduction occasionally, particularly when conditions worsen. Like there's a shift between one generation and another in terms of environmental quality. For others which are purely asexual, I don't actually know, to be honest. It's kind of a question of mine, and I've read occasionally about it, but not enough for it to stick. Like what deals with that accumulation of deleterious mutations? I don't actually know. But sexual reproduction is costly, right? So not only do you only get half of your genetic information into the next generation. Give me a second, John. There's also the cost of reproduction, right? So, if you're a snow leopard, it's the cost of having your children eaten by your new mate. Obviously, spiders have issues. There's always costs that go along with that. So for that to be an evolutionary advantageous process, right, or option, there's got to be bonuses. And those are generally in generating a new collection of alleles, right, mixing the deck essentially for the next generation, so that there's the hope that if conditions change, that generation has, or at least part of them, right, because that mixing is going to be different for each individual. A part of that population is better equipped to deal with that change than the parental generation or even their siblings. Jordan, had a question?
Yes, sir. It's related to the previous one. Not to sidetrack you more, or to sidetrack you with this one, but for asexual organisms, are they less prone, or they're less likely to be affected by negative? I guess you said it was like constant environments. Because what happens if they get irradiated by something? Because doesn't that mean they're just reproducing very damaged cells, and that just feels like it's going to lead into more issues? Or I guess So I guess that environment thing kind of answers.
So in many ways, actually, single-celled organisms, which are a large bulk of asexual reproducers. They're not the only ones. Many insects do too. So simple single-celled organisms actually have many defense mechanisms to deal with external insults like that in terms of radiation, particularly UV and gamma radiation. So they're actually much better equipped to deal with that than we are. So that's one possible kind of mechanism, right, to deal with that. And the second is in more complex asexual reproducers. Many of them have the ability to reproduce sexually under conditions of stress. So they are able to kind of outcross essentially and purge deleterious mutations through sexual reproduction. Even though that has its own costs, the benefits are sufficient that it's worthwhile to do. So it's kind of a trade-off essentially. For really complex organisms like you know, us, obviously there's no way that we would be able to reproduce asexually. It would just be too difficult for that to happen. And so sexual reproduction is the only option. But for others, I mean, you pick the best one basically. But for complex or higher organisms, sexual reproduction, even with all of its costs, has a lot of benefits, right? And so part of that is getting rid of deleterious mutations, so kind of like purging them from the gene pool. And that's also where inbreeding fails, right? Because you're not, you're breeding with genetically similar organisms. And so you don't kind of benefit from that purging effect. So those deleterious mutations tend to accumulate. That's typically the big downside of inbreeding. And also you generate new combinations of alleles, right? Which may be advantageous for the next generation. So that's kind of like the big plus of sexual reproduction. And the fact that most, well, all higher organisms reproduce sexually kind of speaks to how advantageous that is kind of evolutionary over large periods of time. But to do that, you got to have a lot of randomness, right? And that's really kind of the point that I want to make about sexual reproduction. There's as much randomness as possible. So there are different places at which that randomness occurs, right? I'm just going to type out the three of them. Everything is in threes. I don't know why. I actually read an article about why everybody, like journalists, always make like lists of three. Don't know. Anyway. What would we call three sources of randomness? And I'm going to pretend that that's a word. I think it is, but even if it isn't, it'll do. So the first is the random alignment of chromosome pairs in metaphase 2. And we'll get to the details about that. This is just kind of big picture type stuff. 2, recombination between homologous chromosomes. I can't see what I'm typing sometimes. So that generates yeah, that's probably the best way to put it. Novel allele combinations in any given organism, right? And the third is randomness of fertilization. Right, so there's no way to designate which of your sperm fertilize a particular egg, right? It's just fire them all off and see who makes it, basically. So there is a degree of selection for fitter sperm, right? But there's even in that, you know, it's a kind of random as to which one makes it through the eggshell into the egg. Right, so all three of those contribute to generating that random combination of alleles in the offspring, which is different from either of the parents. Right, that's kind of the key. And so as we go through meiosis, you know, I'm going to spend some effort in pointing out where those three different randomness generators, I guess, right, where they occur. And so to be able to have sexual reproduction, you need haploid gametes, right? You can't add 2 + 2 and get what 2, right? So each and every organism that goes through sexual reproduction, whatever the form their gametes take, right? It could be spores in the case of some plants, eggs, gametes, or eggs, sperm are the kind of three main ones. Oh, yeah, good question, Adam. Did I put metaphase 2? Oh, shit bits. I'm sorry. What a dumbass mistake. Hang on. Let me type that back up again. That's a real sloppy mistake. Sorry about that. It's actually metaphase 1. So good question, Adam. Thank you for pointing that out, because that would have really messed everybody up if you hadn't caught that. There's 2 rounds of cell division in meiosis. First one is, you know, interphase, and then you have prophase, prometaphase, metaphase, anaphase, telophase 1, all of them. So what occurs in prophase 1 is very different from what occurs in prophase 2, right? And that's the second round of cell division. And so that's how you go from 1 diploid cell to 4 haploid cells, is through 2 rounds of cell division. First round generates 2 cells, then each of those generates 2 cells through a second round of cell division. But yeah, thank you for catching that. I don't know why I put metaphase 2. Does that make sense? Cool. So essentially, sexual reproduction is 2 parts. One is the generation of those haploid gametes, right, which is results from a reduction in ploidy, right, going from diploid to haploid, shuffling the genetic information around so you have kind of novel gametes, and then fusion of those 2 haploid gametes to form a diploid zygote. And so essentially meiosis is all about generating a big chunk of that randomness, right, which is what parts 1 and 2 in the chat are all about, but also taking a diploid cell and generating haploid cells from that, right? And so essentially almost all of the interesting stuff in meiosis happens in meiosis 1. So basically, meiosis 1 is where it's all at. Because meiosis 1 is called reductional division. So meiosis 1, is 1 diploid to 2 haploid cells. And meiosis 2 is 2 haploid to 4 haploid cells. And that's basically about separating the sister chromatids. So that's called equational division, because you start with a haploid cell and you end with 2 haploid cells, essentially, for each of the 2 daughters from meiosis 1. So meiosis 1, you have both recombination, also called crossing over or synapsis, and you have the random alignment of chromosomes in metaphase 1. Meiosis 2 is really just kind of tidying up the mess, basically. And it's also really important to point out that the daughter cells from meiosis are not identical as a result of recombination. Ooh. Anybody got questions? Okay, so there's a lot going on in meiosis. It's a stepwise more complex and complicated than mitosis. But really, remember again, mitosis and meiosis 2 are more or less the same. Only difference is one is diploid, the other one's haploid. Meiosis 1, now that's where the crazy stuff happens, right? That's where you need to focus all of your efforts in understanding this process. Because that's where the complicated stuff happens. Okay, so just to kind of give you an overview, again, 2 rounds of cell division. Some cells go through a kind of like sort of interphase, but well, some organisms, sorry, go through a sort of interphase between meiosis 1 and 2. Others just go straight from meiosis 1 and, well, M phase 1 essentially into M phase 2. It kind of varies depending on the organism. But you could see here, right, so this is 2n. So n equals the number of unique chromosomes, which are essentially units of information. 2 equals diploid, 1 equals haploid. Jordan.
Hi, sir. I might be asking a dumb question. Equational, it is named that way because question mark. I'm just trying to figure out. Because reduction, it looks like we're splitting, or we're going from 4 cells to 2 in each one of these cells. And then in equational, we're saying half go into one and half go to the other. Is there any, I don't say word trick, but explanation for what equational means in this situation?
So equational is haploid to haploid. So the ploidy, which is how many copies of each chromosome you have. So we're diploid organisms, right? We have 23 unique chromosomes, but we have 2 copies of each of them, right? Those are our homologous chromosomes, right? 1 copy from your mother, 1 copy from your father. So the first step of meiosis is to take your 2 sets of chromosomes, right? So you can see here, this is an organism which is diploid, It has 2 unique sets of chromosomes, and they're different. For each pair, they're different colors to signify from which parent they came from, right? And so now you're going to take one of those, stick it in that daughter, or one of these and stick it in that daughter. And the same for the other one. So you're actually going from having 2 sets of each, well, 2 of each chromosome, however many chromosomes you have, to having 1 of each. So you're reducing the number of sets of chromosomes that you have.
Yeah.
Right? So that's the reductional part. Now, once you've got to that point, if you look at this, it's like, okay, well, sure, I have haploid cells, but I have twice as much DNA as I need because we still have those sister chromatids. So if we go back to mitosis, right, so essentially we have the same, actually, where's a good look? Here we go. We have the same deal in mitosis where each chromosome consists of 2 identical chromatids, right?
Gotcha.
Those are copies that result from DNA replication. And so you have to split those up because you want to have 1 chromatid in one cell and 1 chromatid in the other cell, right? Now, if you go back to meiosis, you have the same issue, right? You can't leave it at this point because even though the ploidy has gone down from diploid to haploid, ooh, sorry, I thought my puppy was going to fall off the couch. You still have 2 identical replicates for each of those chromosomes. Those need splitting apart, right? And that's what the equational part is. Like, you're not changing the ploidy of the cell, like the units of information, right? Because this is 1 unit, 1 chromosome, this is another unit, another chromosome, right? So you're not changing the level of information. What you're doing is making sure you have the right amount of DNA in each daughter.
Gotcha. Okay.
Yes,
Awesome.
that
Thanks,
makes
sir.
And so
Yes, yes.
that's what I mean by meiosis 2 is A, it's like mitosis, and B, it's tidying up, right? So all the important stuff happens in meiosis 1, but you're left with 2 daughter cells which have twice as much DNA as they need.
Gotcha. It just, I was thinking like formulas and whatnot from like calculus and algebra and whatnot. That's why I was a little bit confused on what the word meant. I was like,
Oh, right, yeah. Well, we're biologists. We're kind of sloppy with mathematical terms. Rose. So yes. So if you look at this cell at the top, it has 1 of each chromosome. So it has, if this is chromosome 1 and this is chromosome 2, it has 1 chromosome 1 and 1 chromosome 2. It's haploid. Diploid is having 2 chromosome 1s and 2 chromosome 2s. So we've reduced the number of chromosomes from 2 sets to 1 set. Now, one of the confusing parts is each of those chromosomes, as in mitosis, consists of 2 identical copies. Those are chromatids. Oh, okay. You'll have to put up with my giant fingers and typing. So, So if you did talk about diploid and meiosis 1, right? So that's before anaphase 1, when they split apart. You have 2 sets of chromosomes. Each chromosome consists of 2 identical sister chromatids, right? So after anaphase 1, well, essentially, at the end of meiosis one. That's probably better to put it that way. You have two daughters that are have one set of chromosomes, so haploid, but each chromosome, if I should just copy and paste really, consists of two identical sister chromatids. So one thing that's a point of confusion is that sister chromatids are not, until they're separated, chromosomes. They are the same information, right? So a chromosome is a unique set of information. You can have two very similar chromosomes, those are called homologous chromosomes, right? But they're not identical. So in each of your somatic cells that aren't dividing, you have one set of chromosomes from your father, one set from your mother, right? So you have a 1 through 23 or 1 through 22 in X or Y, 1 through 22 X or Y, right? So they have the same genes, they have the same structure and size, but they have a different collection of alleles because one set you got from one parent, one set you got from the other, right? So they're homologous chromosomes, they're not identical. That's what you see here. So this large pair of chromosomes, the red one and the blue one on the left, that's a homologous pair or a set, right? The difference between that and sister chromatids is sister chromatids result from DNA replication. So they are, one hopes, identical copies. So then you're not adding any information. It's like having two copies of a book, the same edition, right? You're not adding any extra information. You're just adding more weight to your backpack. That's essentially what sister chromatids are. And actually, that's a little bit of a fudge too, because technically speaking, they're not identical sister chromatids anymore. But we'll get into that in a little bit. So then at the end of meiosis, oh, they wouldn't be diploid, Adam, because they're the same information. So that's possibly, hang on, I've just got to move my puppy back towards. Can you see? She's busy shuffling herself off the couch. So to get you just back over. Yeah, there. Yeah, that's good. Stay there. She's like, I was just having a nice snooze. What are you doing? Yeah, so that's arguably the kind of the trickiest point of understanding cell division, right, and DNA replication. So each of a homologous pair, each one you get from a different parent. For every single one of your 23 chromosomes, you have one from one parent, one from the other. That's why we're diploid. We have two sets. When we go through cell division, each one of those, so each chromosome of each set gets copied. So probably the best way, I'll just draw it, be easier. Yeah, don't get excited. So I'm going to draw these as if they're condensed, even though obviously we're talking about cell division. So let's say before DNA replication, we're just going to use M phase as in mitosis, just for simplicity's sake here. Okay, so if you have a N of 2, right? So you have 2 distinct chromosomes. You would have something that looks like this. Right, these aren't joined together. They're just floating around. You can kind of draw them up here if you want. Doesn't matter. We just stick them next to each other because it kind of highlights the fact that they're pairs, right? They're homologous. Right? And homologous basically means same but not identical. Now, once you've gone through the, well, you saw, oh, you missed a shot of the puppy. There you go. There she is. All tired because obviously, you know, it's a hard life being a puppy. So as a result of DNA replication, now you have identical copies for each of, should try and make them identical, don't want one longer than the other, right? So these identical copies are now called sister chromatids. This pair, oh, I'm glad. We didn't spend the whole lecture just looking at her. She's very cute. Obviously, she doesn't do very much. She just mostly lays there and sleeps. Always. Right? So this is still a homologous pair, right? They're not identical because they have some differences between them due to whatever alleles you got from your father versus your mother. But each one of these is now, well, I've stopped her from chewing my power cord so far, although she really likes sandals and flip-flops. So those are now all on top of the cabinet. But each one of those, as a result of DNA replication, has an identical sister chromatid. Now, the role of M phase, at least mitosis M phase and meiosis II M phase, is to generate 2 cells, each with, well, actually, let's not talk about meiosis II because I'm just going to confuse things. Is to separate those into, again, individual homologous chromosomes. So that's the tricky bit, is that these sister chromatids are identical copies that then become individual chromosomes once they're pulled apart. While they're still together, and this is probably the sticking point, Adam, this is one chromosome, this is another chromosome. These 2 individuals do not count as a chromosome, as individual chromosomes, because they're identical. So let me just take a picture. Ah, nuts. I left my Camera in the garage. Just give me a second. I'll put on puppy cam while I go get it. Puppy cam. I think you've been entertaining my class, haven't you? All right, fun's over, everybody. Sorry, can't be 100% puppy. That just wouldn't work. As tempting as that might be. I've never learned so much from... Anyway, all right, so this is mitosis. So this is always the kind of limitations of trying to work off of a PowerPoint. And I've lost my cap for my other pen. Who knows where that's gone? Oh, there. That'll do. Right, so this is mitosis. Now we have, actually want the other pen. This is meiosis. Meiosis, yeah, that's one. One. End of meiosis 1. Now we only have one of each chromosome pair made it into the daughter. If you can see that, hopefully you can. That's funny. What would I do without a puppy, eh? It'd all be like boring genetics. So this is, let's use this color. 2N, because you have 1, 2, 1, 2. You have 2 copies. This is, each of these is 1N, because you just have 1. This might be 2. This would be 2, and this would be 1. Let's say of the different ones. So in mitosis, these homologous pairs are not attached. The sister chromatids are. Those are what get held together until anaphase. In meiosis 1, these chromosome pairs, or the homologous chromosome pairs, are held together until they separate in anaphase 1 to generate these haploid daughters. I'm going to take a picture of that, and then I'll show you what happens in meiosis 2. So we have So at the end of meiosis 2, you have where'd it go? There it is. So I've taken shortcuts using the same color, different color from last time. Right. These are your two. Here we go, that's better. These are your two haploid daughters. And each of these goes through a second round of cell division. So let's say we actually changed things around. Right. So now, and you're going to see the same analogy to mitosis. And this isn't being super accurate because I'm, not changing the plane of division and stuff like that, but it'll do. Each of those now is going to separate the sister chromatids. Exactly, Adam. So this is how you get your 4 haploids. Each of these daughters goes through a second round of cell division to generate 2 more daughters, which go from being haploid to haploid. But now what we're doing is we're separating the sister chromatids. So each daughter has the right amount, like weight, literally, of DNA. And so now you end up with this. And that's one cell. This will be another one. This will be your third one. And this will be your fourth one. Each of those will be haploid. So meiosis 1 is all about separating the homologous pairs. So now you only have one of each homologous pair. So you go from one diploid, which is kind of over here in my couch cushion, to 2 haploids. That's the reductional division step of meiosis 1. So your original gamete was a diploid cell and you start with 1. I'll write this out in a little bit once I turn the computer around. You generate 2 haploid daughters, but each of those still has 2 sister chromatids for each of the 2 chromosomes it has. Meiosis 2 is all about separating those sister chromatids so that you have 2 daughters, each with 1 chromosome each for each chromosome. So that's why this part, meiosis 2, is equational, because you go from haploid parent to haploid daughters. Haploid, haploid. Meiosis 1 is reductional because you go from 1 diploid parent to 2 haploid daughters. Yeah. So I'll write that down again because it's important. Make sense? That's why it's so important to be careful about what stage of meiosis you're talking about. Meiosis 1 versus meiosis 2. Because they are quite different. Okay, so I'm going to rip on a little bit because I've got other stuff that we need to cover. So get the chat back up. So basically the two most important things that happen in meiosis, generation of gametes, not talking about sexual reproduction as a whole, but in the generation of the gametes, occur in meiosis 1. So the first thing that happens is that we have recombination, right? So recombination is basically a series of steps that occurs throughout prophase. And as you can see, we just have prophase here. We're not distinguishing it by prophase and prometaphase. It's just kind of as another example, rows of, you know, how they aren't necessarily distinct parts. So hang on, let's get this back wherever it's supposed to be. Zoom is such a pain sometimes. So in prophase 1, the homologous chromosomes, I'm just going to call them chroms for speed, align using what's called the synaptonemal complex. And basically what that does is it base by base, you have chromosome 1 from mother, chromosome 1 from your father, right? So they are homologous pairs, not identical, but homologous. They are aligned base by base. It's actually quite astonishing, right? How accurate this is. As those chromosomes condense. And the reason is, and really it's actually, it looks like this, right? Because we've got, this is chromosome 1 from father, chromosome 1 from the mother, right? So these are the sister chromatids, chromosome, right? They get aligned like so, right? Base by base, literally like knuckle by knuckle. I think I've got that pretty well mapped out. As they condense down into those kind of super condensed, you dog are such a pain in the butt. Hey, love, could you rescue Puppy from the couch? She's digging. Oh, no, maybe she's settled down. Anyway, and as those chromosomes condense, they're going to be lined up perfectly. The reason for that, oh yeah, or rescue the couch from her, I guess it depends on your perspective, is that we're going to swap parts of this chromatid with this chromatid, right? So these are non-sister chromatids. They're not identical, right? We're going to line them up, and then we're going to basically take a little bit of this, and we're going to stick it onto the other one. I don't know, she's settled down now. She is just trying to dig through the bottom of the couch to find what was underneath, I guess. Right. And the purpose of that is to create what are called chiasmata. Right. So then kind of mid to late prophase 1, parts of each non-sister chromatid are swapped via what's called a chiasmata, or chiasma in the singular. And that generates chromatids that are different, basically in terms of alleles, which is what we care about, right, in terms of genetics, from either parent. Right, so some of the chromosomes that I have in my cells look exactly like my father's or my mother's. Some of the chromosomes I have in my cells are combinations of both as a result of recombination in prophase 1. So I don't know, let's say chromosome 8, I might have a little bit of it that is from my mother and the rest of it from my father, or vice versa. This is like the fine scale shuffling. It's like rather than shuffling a deck of cards, instead you're taking a bit of, you know, the ace of spades from one pack and you're replacing the exact same part on the ace of spades from another pack. So it's not large scale shuffling, it's very small scale chromosome level shuffling. So that recombination is one of the ways that we generate, or meiosis, generates kind of or randomly generates even, new allele combinations. Also, these crossing overs, these chiasmata, actually help hold these homologous chromosome pairs together until metaphase 1 checks occur. And so the reason is you have two kind of sets of cohesin. Not cohesion, thank you, autocorrect. One set on the arms, right? Actually tips of the arms. And one set at the center of the chromosomes. And so this is how we control which bits of DNA get separated. So in metaphase 1, again, we're going to have the chromosomes as pairs lined up on the spindle, on the metaphase plate. Now they're held together by essentially, it's kind of hard to show with fingers, unfortunately, right? The swapped over bits are still stuck to their sister chromatids at the tips. Mostly just the names and the order in which this stuff happens. Right, so the synaptonemal complex disappears after crossing over occurs. The chiasmata remain until anaphase 1, because they're actually what hold together the homologous chromosome pair. Oh, and Rose, I will answer your email. I just had a crazy day yesterday and I didn't have time to do so, but I will do. And so in metaphase 1, right, those chiasmata basically hold the chromosome pairs together, right? And they do that because essentially, let's just keeping my eye on the time. I don't want to run out of time. If you look at a chromosome pair, see, this is the downside of puppy time. We have less of this time. So if we draw this up really large, what you have is this crossover like so. Like that. So we have, this isn't an ideal diagram because it's actually a little bit more complex than this. But this crossing over holds together these two chromosomes. So when you go through anaphase, we still have cohesin holding these sister chromatids together. We will no longer have the cohesin that holds together the tips. And so that allows us to pull apart this homologous chromosome pair. And then obviously we get the division going down like so. So at the end of meiosis 1, each cell has one chromosome each. But the sister chromatids on each chromosome are no longer identical, because these bits were originally identical, but now the ones that experience recombination now have a little bit of the chromatid from the other chromosome. Right, so if you think about this in terms of alleles, if we just do it super simple like so. I'm going to use D. C as a gene letter absolutely sucks. Now we have our parental chromosomes, which look exactly the same as you would find in the parents. Now we have a non-parental pair. Right, so now we have a new combination of alleles that wasn't present in either of the parents. So that's one central way in which we generate new combinations of alleles in the gametes. The other way is, let's make sure we've got the chat back up. Oh, let's just finish that train of thought first. I don't know why the N is capitalized. So that's just continuing that kind of thought that I had earlier about this is how you control which bits fall apart when. And we get into, yeah, interkinesis is kind of weird. Happens in some cells, doesn't happen in others. Basically, it's the interpart between meiosis 1 and meiosis 2. Oh, and also, where do we have it down here? So this is slide maybe an oversimplification, but it's actually a really important one. Okay, so here is a good example of what we're talking about in terms of the cohesion of different parts of the chromosome. But I'm sorry, I'm kind of a little flustered because we're running out of time. So the other way in which randomness is generated is through the random alignment of chromosomes on the metaphase 1 plates. And so here we've got 3 homologous pairs, right? So this is diploid because there's one of each, right? So this is 2n when n equals 3.
So you're not going in order, right? You're just telling us anything like of importance.
Oh yeah, so that's a kind of common thing from when I teach this stuff kind of online, even though the PowerPoint contains all the information you need to know, but depending on what questions come up and what trains of thought I have, I'm not going to go through it linearly.
Okay.
Yeah, so that's kind of an unfortunate downside of having the PowerPoints already prepared, in that I don't always teach every class the same way. It just varies depending on kind of what stuff comes up. Does that make sense?
Yeah, that makes sense. I was just getting kind of confused, but I understand now.
Yeah, that's the downside, right? So hopefully it kind of will make more sense when you look back over it. But also sometimes I just forget stuff that's important and then I have to go back and go, hang on, this is really important. You know? Okay. This puppy has a habit of trying to jump off the couch. So
yeah, because I was looking at this a lot, but like I think the homework, it's like kind of, I don't know why I had so much trouble with the homework. Like it's like it went with the textbook more, not
right.
the PowerPoint, which is okay.
Yeah.
I couldn't find any of the answers yesterday.
So sometimes
the
homeworks are pretty heavy on kind of terminology like language and stuff like that. Whereas I tend to focus, although I obviously need to give you that, I also try and focus on the kind of like the concepts and principles that are at play. So the homeworks are really a kind of like a complement, I think is probably the best way of describing to the lectures. You know, they don't, one doesn't replace the other. They just kind of, it's basically another way of getting you that practice with the language and some of the terms and some of the principles too, but a lot of language type stuff. Does that make sense?
Yeah, no, that makes perfect sense. You'll just have to use all of your resources that you have, not just the lectures, not going off that. So, okay,
Right,
perfect.
exactly. Yeah, I mean, it's kind of all of it, right? And just the PowerPoints alone aren't going to help you either, because there's stuff that I go through in more depth in class than it's gone through in the PowerPoints. And so the hope is kind of, it's like a kind of complete package, essentially. So anyway, the other, got my eye on Puppy. Puppy is back. Hey, here, chew on one of those instead. Here we go. I'll have to fish that back in a few minutes. That's the only thing I've got to hand. Stop her chewing on my laptop power cable. So the other thing that's really important, and this is really the basis of the law of independent assortment in many ways, sorry, is it's totally random which way round that chromosome pair aligns on the metaphase 1 plate. So for chromosome 1 up here, right, we've got the two pairs. It could be either this way, or it could be this way around. It's totally random which way round it's going to be. Similarly for chromosome 2, it could be this way, or it could be that way. Each individual chromosome pair is totally random which way round it will align on the metaphase plate. And so the outcome in terms of what you get in the gametes is a random assortment of chromosomes. You could have a maternal chromosome, chromosome 1 in one gamete, and the paternal one in a different gamete. But that is random for each chromosome. So the more chromosomes you have, the more possible combinations you can have in your gametes. And I think in humans, it's 8.4 million different possible combinations of chromosomes. And that doesn't include recombination and like that small scale shuffling that you get through crossing over. So identical twins and triplets are identical. I don't actually know the mechanics of why identical twins occur, but they will have exactly the same DNA, which is why they're identical. So everything that I'm talking about here doesn't really apply. And I think it just depends on when during gestation that funky division happens, whether or not you're identical or non-identical, something like that. I really don't know much about twins, to be honest. Okay, so again, here's, we've got lab in a little bit, snooze. Here's a good, actually I've led you somewhat astray. I forgot Shigoshin is only in meiosis, right? Not in mitosis. Sometimes I just get my wires crossed. I'm sorry about that. But here's a really good breakdown of the center of the arms versus the tips that allow the homologous pair to separate in anaphase 1 and the sister chromatids in anaphase 2. What? And this is just a good summary of the differences. Yeah, she needs something else to chew on other than my bloody hell. Look at that. Puppies, eh? Yeah, she is most definitely teething. She's a little pain in the ass. So the big points to get across mitosis is generation of two. Oh, you got something else, Richard? Sean? Good stuff. Two identical diploid daughters from one diploid cell. Meiosis is generation of 4 non-identical, give me a second, Adam, daughters from 2 rounds of cell division, those daughters being haploid. No, the kinetochore is the complex of proteins that the spindle fiber is attached to. Cohesin is the protein that holds the sister chromatids together. And so that protein is really important because it prevents, in meiosis 1, it prevents the homologous pair being pulled apart. In mitosis and meiosis 2, it prevents the sister chromatids from being pulled apart. That's the role of cohesin. And that ties into the spindle assembly checkpoint and the anaphase promoting complex. So the kinetochore is very much like a docking socket, which the spindle fibers attach to and exert tension on the chromosome. Cohesin is a glue that sticks the sister chromatids together. And that glue has to be broken down to allow anaphase to occur. Okay, I think I managed to cover everything here. The other thing from meiosis is that meiosis 1 generates the non-identical, well, yeah, is the source of the non-identical daughter cells. And the non-identicalness there, such as it is the result of both recombination, so the generation of new kind of versions of the chromosomes, essentially, or new combinations of alleles that weren't in the parents, and the random alignment of the chromosomes on the metaphase 1 plate. Those two things together will generate 4 daughters that have different mix of maternal versus paternal chromosomes and parental versus recombinant chromosomes. And obviously when those gametes join with a gamete from another individual, the opposite sex, then you're going to get all of that randomness and all of that randomness together to create even more randomness, right? Newer combinations. That's why you look a little bit like both of your parents, but also different from either of them. Because essentially some of the alleles you got are just from, well, I mean 50-50, but the combination varies for each chromosome. Does that make sense? I mean, it will always be 50-50, but which bit of the 50 you get from which parent is going to change. So hopefully now you can see the very marked difference between mitosis and meiosis. But meiosis 2 and mitosis are very similar, because it's all about the separation of the sister chromatids. Meiosis 1 is very different from either of those. Okay, does anybody have any questions? Probably all brain blasted by now, and my ears ache. Although it is my daughter got me something to occupy a little bit. Okay, so we have, well, the exam is now open. So it's free for you to take until midnight tomorrow night, Friday night. You have 75 minutes to take it unless you have a letter of accommodation from DSS. Oh, we will have lab in half an hour, Dipper, once I've scarfed down some lunch. But yeah, I'm sorry. It's what we have to do, right? But you're able to take the exam at any time after now until midnight tomorrow night. Just don't leave it too late tomorrow night in case any problems occur. You've got 75 minutes to do it in. Don't rely on it being open book to get you out of a hole. There's going to be stuff you're going to have to calculate, chi-squared. You're going to have to do some monohybrid, dihybrid test crosses, and obviously there'll be a bunch of stuff about what we've just learned today. So I'm more than happy to answer questions, obviously not now because I really need to eat my lunch, at the end of lab, and also by email, you know, anytime between now and tomorrow afternoon. So I will be, hang on a second, Adam, I will be going out of town probably mid-afternoon. So if you have questions, please try and get them to me before then. Otherwise, I'll be out of contact. So terminology and math, I'd probably say, oh yes, I will definitely, Cameron, upload those probably straight after lab. So they'll be up like between 4 and 5 o'clock this afternoon. Awesome. Adam, it's going to be a mix. There's going to be a lot of math because you're going to need to work out different things based on the principles that we've learned in terms of Mendelian inheritance and probability and statistics, right? So there's going to be a bunch of that. There's going to be a bunch of just basic recall stuff. And then also kind of, you know, figuring out different things based on the stuff that we've learned. It's going to be a mix, essentially. So there will be stuff that you'll be able to answer the questions, you know, instantaneously almost. There are going to be other ones where you're going to have to spend, you know, 5 minutes or so at least, working out something, right? So those questions, even though they have equal points, don't necessarily have equal difficulty or equal time kind of associated with them. Does that help? Cool. But again, any other questions you got, we'll have time at the end of lab to go over that. It won't be too long of a lab today. All right. My daughter's toasting her lunch, which is making me hungry. So I'm going to head off and I will see you in 30 minutes. So at 10 past 2. Bye-bye.